I think that given the interest of time, I think we're going to move on to the next wonderful speakers. The third speaker is our Senthil Muthuswamy. He works in the Beth Israel Deaconess Medical Center, and he's the guru of all these organoids that we're very excited to hear about. So Senthil's going to give us a talk on trickling T cells into action using patient-derived organoids to identify and expand tumor-specific T cells. Senthil, please.
Okay, thank you. I hope everybody can see the slides properly. Can somebody confirm the slides are okay? Okay, all right, I'm going to proceed. I would like to thank the organizers.
Yes, looks good, at least on Zoom.
Okay. I'd like to thank the organizers and Alana and Laura for the invitation to speak in this session. I will focus our talk on studies related to organoids in immuno-oncology. In terms of disclosure, I have one. I'm a board member for CAR Medical. The overall research program in my lab really spans models and mechanisms theme. We generate models, mostly cell culture-based models, whether it's 3D culture or organoid models, to study cancer biology and cancer translation research. From a mechanism point of view, we focus on cell polarity pathways that regulate either cancer biology, metastases, or therapy resistance. Now, for today's talk, I'm going to really focus on the models arm. And my interest in development of these sort of culture methods started when I was a postdoc a long time ago, when we developed a 3D culture platform using human mammary epithelial cell line, MCF10A. And we use these cell lines for various aspects of cell biology studies and modeling cancer genesis. And I think as many of you may know, this platform is quite widely used in the breast cancer community today. Now, from this, more recently, we've been more focused on developing patient-derived organoid models, usually tumor-derived or sometimes stem cell-derived. And the reason for this is that because I think it is important to be able to not only grow cell lines in culture, but also primary patient tumor cells. One of the key aims we have, or the goals we have, is to develop conditions specific for each tumor type, which is devoid of any of the stem cell factors. And I think key criteria there is to make sure that we maintain the differentiation status and the histoarchitecture of the patient's tumor as close to it as possible, though growing in culture. And with that, I'll give you an example of what I mean by that. Here are the two examples from pancreatic cancer, head and neck. What you notice on the right-hand side is that the histomorphology of the cells growing in the culture conditions are actually uncannily similar to what is seen in a matched patient tumor. So we have used these type of platforms for various studies, and I'm just going to highlight two recently published studies this year and late last year. Just highlights the power of using these patient-derived tumor models for translation research. The first one, we demonstrated that the organoids have more than 85% accuracy in predicting in vivo drug response in the context of PDX tumors. And the second one that we just recently accepted in clinical cancer research demonstrate in a co-clinical setting that organoids generated from patients with pancreatic cancer are able to predict drug response of the matched patients with almost 80% accuracy. These two studies really demonstrate the power of using these patient-derived models as a tool for modeling patient biology, tumor biology. But for the sake of today's discussion, I'm going to really focus on why we think organoids are a platform for immuno-oncology space. In this next couple of slides, I'll actually explain the reasoning and logic for why we think so. The first one, as I mentioned, that the organoid platform maintains the differentiation status of the patient tumor, which is very important because you want, if the tumor cells are being targeted by T cells, you want to maintain the tumor cells in its native state as possible. And as many of you may know, and also you heard from Jennifer earlier today, that the organoid platform does in fact are quite effective in maintaining or at least conserving mutation and gene expression profile of the matched patients. An important criteria that we recently demonstrated is that organoids, in fact, preserve the cell surface glycan profile as an in vivo tumor with more than 85% accuracy. This is extremely important if you think about it for a minute, because the T cells see the tumor cells on the cell surface, and many of the surface proteins are glycoproteins. So the fact that we are able to maintain the glycan profile speaks to the volume in terms of the antigens that are being presented by these cells in culture. So then the question then is, really, why do we need, we developed a co-culture platform, and the question is, why do we need a co-culture platform for these things? I think the answer is threefold, because as many of you may know, that ER-positive breast cancers are actually so-called cold tumors, meaning there are very few tumor-infiltrating lymphocytes. So in that setting, how do we really understand how T cells can see those tumors, or do they not see at all? I mean, that's a question that we need to understand. The second is that the T cells that are present in the tumor bed, not all of them have the cytotoxic property. It's only a fraction of them can actually target and kill the tumor cell. That means many of the T cells we see as TILs are actually guilt by association, meaning they're just present there for the sake of, by whatever chances they are. And because of this, I think if we find a way to identify T cells that have potential to kill tumor cells quite effectively, that may enhance our ability to find and understand T cell mediated killing. The third reason is also is that most of the tumor antigen discovery efforts today is really based on in silico methods. Whether it is a new antigen prediction from mutations or gene expression profile, it really depends on our ability to predict what peptides will be presented by the MHCs. We think that coming up with an empirical way to really find T cells that target, can kill tumor cells, and using them to discover T cell receptors and antigen might be really powerful and game-changing. So with that idea in mind, we developed a platform where we can grow organoids from patients, and from the same patient, we collect peripheral blood, and we put them together in a co-culture platform. And we allow the organoids to educate and also amplify the T cells that are present in the peripheral blood mononuclear cultures to generate a population of T cells that have the ability to kill tumors. We call them as organoid prime T cells, or OPT cells. Now, the platform works beautifully. You can take organoids combined with PBMCs in the same patient. In about 14 days, a population of T cells can now attack and kill the organoid, and in the process, they also expand. And when we analyze these organoid prime T cells by site-off analysis for surface markers, what you will notice is that many of these tumors, every patient has its own, oops, I'm sorry. I'm sorry, I lost my presentation. Okay, sorry, got it back. We found that every patient has its own sort of T cell repertoire. The patient 3, for example, is primarily made up of CD8 positive T cells, whereas patient 38, for example, are made up of CD4 positive T cells. So and this actually opens the possibility that by this co-culture method, we will be able to find different types of tumor targeting T cells. Now, these T cells are in fact cytotoxic, because if you take a green labeled organoid and co-culture them with the OPT cells, by about 8 to 10 hours, you can see that they're beginning to attack the organoids, and by about 20 hours, the organoids are completely obliterated. And if you analyze these T cells, they actually show all aspects of cytotoxic properties. They respond to the stimulation by increasing proliferation, they secrete interferon gamma, and they also secrete granzyme B. As you would expect for a cytotoxic properties. Now, as I mentioned, some of them are CD4 and CD8 positive T cells. When we dig deeper into the mechanism by which they kill, we also find that there are different mechanisms by which these T cells kill the tumor organoids. For example, the CD4 positive T cells if you co-culture them and measure the levels of epithelial keratin in the media, it demonstrates that OPT cells, in fact, induce killing as monitored by increase in the keratin levels in the media. However, the interferon gamma levels do not change. Now, if you look at as you know, there are other mechanisms by which T cells can kill. And in fact, it turns out that the patient 38 CD4 T cells have a higher levels of Fas ligand. And if you now interfere this Fas ligand by using a blocking antibody, the blocking antibody does not interfere with patient 3 mediated killing, which is CD8 positive T cells, whereas it actually blocks patient 38 mediated killing. This is CD4 positive T cells, demonstrating that we in fact have the ability to find different types of tumor targeting T cells in culture by this co-culture approach, and opens the possibility to study them better. A really interesting aspect of this whole co-culture method came when we analyzed the clonal makeup of these OPT cells when compared to the PBMCs. And the way we did it is to generate DNA from PBMCs and OPT cells, and then sequence the TCR beta in this, and then analyze what percentage of TCR beta of a given TCR receptor is a representative of the entire population that we have with sequence. The results are quite striking. For the PBMCs, for example, no specific TCR represented anywhere from 2 to 5% of the overall population, suggesting that this is a highly polyclonal population of T cells, which is what you would expect. However, OPT cells, some TCRs were represented for about 90%. In general, 5 clones were sufficient to make up 99% of the TCRs that we sequenced, suggesting and demonstrating that there is thousands of fold expansion that we observe when we put these cells together. They're identifying as this platform as a potential way to find specific TCRs that have the ability to actually attack and kill the tumor cells in this context. Now, the real power of these TCRs can only be seen if you know that these TCRs are transferable for killing abilities. So the way we decided was we actually identified both the variable, the V alpha and the V beta variable chains, and made a chimeric TCR using mouse constant regions expressed in heterologous T cells, and asked, can these heterologous see and kill tumor organoids? And the results were quite beautiful. It turns out that if you take a patient 10 derived OPT cell TCR and then co-culture either with patient 10 organoids or patient 38 organoids, what you notice is that the interferon gamma response is prominent when you co-culture with autologous organoids, but it is less prominent when you do with allogeneic organoids. And I think this weak response is probably due to the allogeneic response. And more importantly, this response is dependent on MHC class 1 antigen presentation, because if you use a pan-MHC class antibody, you can block this TCR-mediated killing of the suggesting that in fact, these heterologous TCRs, these TCRs expressed in heterologous T cells retain MHC class-dependent killing of autologous tumors, demonstrating the potential for these TCRs to be TCR therapeutic approaches. Now, all the studies I showed you so far were actually platform building exercises that were done in the pancreas cancer setting. Now we have moved this into the breast cancer setting, and we have now actually have quite beautiful studies that has revealed some really interesting and new insights that we didn't expect before. We initiated these studies using mouse models of breast cancer because this allows us to be able to do both in vivo and in vitro studies in an immunocompetent background. And we used 2 models. One is to model ERB2 positive breast cancer or HER2 positive. Another one is to model a luminal type of cancer, which is polymovirus middle T oncogene-driven cancer. So we generated mammary tumor-derived organoids and matched a syngeneic normal mammary epithelial-derived organoid cultures. And we did the co-culture of T cells derived from the splenocytes of these same mice. And what we found was that when you look at patient 3, we found a lot of CD8 positive T cells that were selected in the OPT population. When we analyzed the TCR makeup, most of them express the alpha beta TCR. That is the most common type of TCR that we talk about usually. And what we found with, by analyzing these, we found 2 things. OPT generated from one mouse can kill polymovirus middle T tumor from different mice, which is what you sort of expect because they're syngeneic tumors and they may share antigens. So polymovirus middle T, 3 OPT cells can kill 4 and 5. The second point is that the same OPT cells did not have any interferon gamma response in response to normal mammary epithelial cells from the same mice, suggesting that whatever they are seeing is actually present in the tumor epithelium and not in the normal epithelium. We did the same experiment with ERB2 positive tumors, these tumor organoids and normal organoids. And here came a bigger surprise. It turned out that when we looked at OPT cells from these new mice, more frequently than not, they were double negative T cells, CD4 negative and CD8 negative. And they were mostly expressing gamma delta T cell receptor and not alpha beta. This raised sort of interesting questions as what does this really mean? And for us, it was actually an educational moment because we were really not thinking too much about gamma delta T cells. So let me just send one slide for those of you who are not familiar. Both alpha beta and gamma delta are actually derived from the same precursor in the thymus. They both are cytotoxic T cells. Both produce inflammatory cytokines like interferon gamma. They differ in very important aspects. One is that there is the receptor rearrangement for gamma delta is limited, and this is less repertoire compared to alpha beta. And the second most important difference is that whereas alpha beta sees antigens presented by MHC antigen presenting molecules, gamma deltas do not use these antigen presenting molecules. And this has two major benefits. One is that because they actually see antigens in an independent manner, they don't have a problem with HLA mismatch issues that come with going from one patient to another. So these gamma delta T cells can go across patients quite smoothly. The second is that MHC downregulation is one of the immune evasion mechanisms that many tumor cells use. And this is not applicable for gamma delta T cells. And the third point, which actually can be interesting in the long run, is that the gamma delta T cells can actually see non-peptide antigens, phospholipids, and so forth. As some of you may know, that tumor cells, in fact, differ significantly from normal cells in their glycan profiles and the surface glycolipid profiles. So whether those are actually antigens seen by gamma delta T cells is something that is not really fully understood. And from a clinical point of view, it does look like this has actually an understudied population of T cells. So this study was published a few years ago in a pan-cancer analysis of 18,000 tumors with overall survival. The CyBER sought for what type of T cells are present in a tumor gene expression profile and how does it relate to the survival of these patient tumors. The data actually, I just want to make one point in that if you look at all the types of T cells that were deduced from this data analysis, the top cell population that correlates with favorable outcome for multiple malignancies is gamma delta T cell population, suggesting that there is something positive about having higher percentage of gamma delta T cells in the tumor milieu for a good outcome. And exactly what they are seeing, how they are killing, are questions that are of great interest to a lot of people in the field, including us more recently. And there has been a number of studies that are currently ongoing, multi-clinical studies and so forth, that really are focused on expanding gamma delta T cells in culture and then giving it back to the patient. And because there is no specific way to expand tumor targeting T cells, most of the methods use non-specific agents like immunobisphosphonate. The success has been fairly limited. So we are excited by what we see because we think maybe this co-culture approach could be one way to enrich for tumor selective gamma delta T cells. And that's something that we're keenly interested in. So as I mentioned, the gamma delta T cells see antigens independent of MHC presentation and can also be targeted for multiple different types of tumor types. So we wanted to know if gamma delta T cells for one mouse model can kill tumors from another mouse model. So the way we tested is to grow cell lines from either polymovirus middle T or new model, and then we co-culture them with OPT cells and then look for viability or killing. What we found was that the new derived OPT, the new ERB2 tumor-derived OPT cells can effectively kill both middle T tumor cells and new tumor cells. These are three independent mouse tumor-derived cell lines. But it has no effect on the normal mammary epithelium, whether it is derived from a polymovirus middle T mouse or from a new mouse, suggesting that it is seeing something tumor selective that is shared between the two antigens. And what it is, we don't really know, but something that we are keenly interested in. And this is just a quantitation to show, in fact, that you can quantify viability, and it is quite dramatic in killing. One important point I wanted to make is that if you notice in this pie chart, there is 16% of the cells were alpha beta and about 81% were gamma delta. We wanted to make sure that this is not, the killing we see is not residual effect of this alpha beta. So we enriched for gamma delta population, so we have 99% purity, and then retested them, and we demonstrated that in fact pure population of new gamma delta cells are in fact cytotoxic in this context, giving us further confidence that this is something we should worth thinking about. And so we wanted to know if this works in vivo, and the way we did it is to take a luciferase expressing mice. This is done in collaboration with Lori Littlepage at Notre Dame. And so we generated orthotopic transplant mouse, injected these OPT cells, especially from this gamma delta cells, and then followed by imaging. I'll add a caution, this is early studies, and we are repeating this now for a larger cohort, but it is exciting, the initial results are exciting. In the control case, we see tumor growth over a period of 24 days, whereas in the context of an OPT injected cells, we see tumor shrinkage over a period of the time period we looked at. So we are now excited that maybe this is relevant and interesting in the context of in vivo in the syngeneic mouse models. We're also now expanding this into the human setting. It's a collaboration with Alana Welm's group, where we generate organoids from the existing PDX tumors and also use archived PBMCs of the same patient that gave rise to that PDX tumor. And we have now generated populations of T cells that can actually be co-cultured with organoids, and then we can characterize them. And we did this for one of the patients, this is patient 39. These patients now, we are able to generate a population gamma delta TCR expressing OPT cells from this patient 39. And interesting early results suggest that if you now take this patient 39 OPT cells and then co-culture them with patient 32 organoids, this should actually read 32. What we found is that this patient 39 derived OPT cells can kill 32 patient 32 derived T cells organoids, suggesting that it has the ability to kill multiple patient tumors. Whether this pans out in vivo or not are experiments that we need to do. So with that, I will stop here and conclude saying that we think this co-culture platform is actually quite powerful, especially for breast cancer. We may be able to use OPT cells directly for cell therapy, but more importantly, I think this is an empirical platform where we can identify either anti-tumor targeting TCRs and or even T-cell tumor targeting antigens. And more importantly, perhaps these TCRs can become tools for TCR therapy approaches that can be broadly used for cancer, blocking cancer progression and other things. So I'll stop here and thank the people who do the work. So almost all the work I talked about today is done by two talented postdocs in the lab, Xinda Meng and Shang Shan, and in a strong collaboration with Steve Elledge's group on a lot of the TCR studies, Lori's group on some of the in vivo experiments, Lena's group on the PDX models. And I didn't have time to talk about Jens Yonkers and Nelly Polyak's group on rat models, and John's group for some of the site-off analysis we did. And I'll stop here and take any questions.
Thank you, Senthil, for a wonderful talk. I think I see a couple of questions are coming up in the Slido. The first question is, what is your negative control to determine your OPT specificity? Like, for example, we think that these OPT cells were generated from, individual patient, and how can you tell, like, what is your measurement to say that this is specific to that cancer-oriented organoids, which was primed from, or versus, I know that you have shown some activity against some of the other cancer models, but how do you actually measure your specificity, sensitivity compared to some of the other more generalized model?
So if I understand your question correctly, you are asking whether it is specific to autologous tumors, or does it actually attack multiple patient tumors? Is that what you're asking, or normal versus tumor?
I think just among, if I understand the question correctly, I think among all the other patients. So what is the specificity for that individual patient?
So I think we have tested this. So for example, if you look at breast tumor-derived OPT cells and we ask whether it kills pancreas tumors, it doesn't, from that point of view, for different patients. And between patients, it looks like in most of the cases when we generate OPT cells, if it is alpha beta TCR expressing CD8 positive or CD4 positive T cells, its activity is more specific to autologous tumors and not to another patient-derived tumor, whether it is the same breast cancer or pancreas cancer, for example. In terms of normal versus tumor, there the data is weak in our hands. We know we have strong data in the mouse model setting because we can test it for normal killing. In terms of normal cell-derived organoid killing, in terms of patients, what we have done is that we have, in some cases, especially for pancreas cancer, we have tumor-derived stroma, and the stroma is not attacked by the OPT cells, whereas the tumor cells on the same patient are attacked. We can also grow sometimes B cells from the same patient, and we have tested them, and it doesn't attack B cells. We don't have a spectrum of normal tissue from that patient to know if it is tumor specific, and that is a big challenge we have, and I think it's a challenge in general in the field.
Thank you. I think the next question is, how long these OPTs last? For example, would they eventually develop the resistance, through the organoid models?
Yeah, so it seems to vary between patients. For some of the patients, we have kept them in culture for months, and they still have the ability to attack the tumor organoid quite effectively. But in some other cases, they do slow down in proliferation after about 5 to 10 passages. And a hidden question there could be that if you now repeatedly stimulate, do they actually show exhaustion phenotypes or not? We think they do if you don't give them time to recover. If you just continuously challenge them, they do seem to slow down the killing after 2 or 3 rounds of killing, suggesting that they do show signs of exhaustion and repeated settings.
Thank you. Stan Lukowitz from NCI is asking, can you clone and put the gamma delta TCR into naive T cell and then get the similar responses?
Can you repeat that again, please?
Can you clone and then modulate the gamma delta TCR into a naive T cell and then get the same responses?
Yeah, great question. So we are now currently, the last couple of slides I showed is the first piece of data we have for human gamma delta T cells for breast. So we are now validating those experiments and doing the TCR sequencing to be able to generate a chimeric TCR. And that's something we're actively going to do very soon.
Wonderful. So I have one more question here, and then I think we have a one in live question. So Eric is asking, thank you for the great talk. Have you found a common anti-tumor TCR that is selected among different PDOs that you have studied, or are the anti-tumor TCR is more highly tumor specific from your observations?
Yeah, so far from our observation, it's tumor specific, but I could not say for sure because to directly answer the question, you need to have enough samples to analyze. So we have only a limited number of organoid and OPT cells that we have analyzed so far. So the real answer will come if we were to do a panel of, let's say, 25 to 50 organoid and OPT cells, and then analyze them, then we'll be able to answer that question. I certainly hope that if you analyze enough breast cancer samples, we will find some things that are shared between patients, but we don't have the data to support that yet.
So Alana needs to send more samples, I think. And then I think there was the question in the audience. It's answered? Okay, all right. So I guess